transcription. We finished that. Now we're moving on to translation. We said that the tRNA is what carries both the anticodon, which is the nucleotide sequence, and the amino acid. So the tRNA is really the point at which we are translating from nucleotide language to protein language. So each tRNA links a particular mRNA sequence or codon to a particular amino acid. The tRNA anticodon matches with mRNA codon. Like mRNA, tRNA is made in the nucleus. The tRNA is used over and over again. So again, this is what it looks like. It's looped like this. It kind of looks like a T as well. This is the anticodon region, which comes, let me draw this again, which comes and pairs with my mRNA, which has these codons on it in groups of three. So this codon on my mRNA is going to pair with the anticodon on the tRNA. So my codon on my mRNA is going to be UUC to match with AAG. So that's how it works. That's how you go from mRNA to the amino acid, which would be attached up here. If you know the DNA code, you can find the mRNA code and then the amino acid sequence. So this is my chart, the amino acid chart. Now, RNA does not contain thymine. Instead, it has uracil. U binds with A, code is universal and redundant. So these two words are important when you're answering long answer questions. You want to use keywords that tell the person marking your work that this guy knows what he's talking about. What does it mean if a code is universal? It means every organism uses this code. So whether you're a bacteria or you're an elephant, you use the same code chart. As in a bacteria, a triple U codon will code for phenylalanine. A CAA codon will code for glutamine, etc. Okay? And what does it mean if a code is redundant? Redundancy means that multiple codons will code for the same amino acid. So for example, this one is a good example, arginine. We have 4 codons that code for arginine. If you look closely, these are not random codes that code for arginine. These codes all have in common CG as the first 2 nucleotides. So CG, and then the last position here, that's where the redundancy comes in. Whether it's a U, it's a C, it's an A, or a G, doesn't matter. It's these first 2 that practically determine the amino acid. So redundancy comes when you have 2 or more sequences coding for the same amino acid. Basically, like when you think of the definition of redundant, it's like you have one code coding for arginine, why do you need others? So redundancy is when you have multiple things doing the same thing. Multiple codons for same amino acid. That's called redundancy. And actually, redundancy can decrease mutations as well. So not exactly decrease the occurrence of mutation, like the frequency of mutation will stay the same, except, for example, if I have in my DNA sequence, I have CGU or RNA sequence, CGU, and my U is mutated to some other nucleotide. Let's say it's mutated to G. Guess what? I'll still have the same amino acid being coded here. So this mutation will actually not make any difference in my life because my end product, which I care about, which gives me the phenotype, which again I care about, it's still arginine. It is the same thing. So when we have redundancy, we reduce risk of phenotype change as a result of mutations. Let's say I'm going to call them deleterious. Deleterious means like kind of like lethal or very extreme phenotypes. So we reduce the risk of that because we have redundancy in our code. Meaning, if I mutate any of these, it actually gives me the same amino acid. It makes no difference in my life. Okay. So this dogma, we call this the central dogma. Remember, going from DNA to mRNA to amino acids. So you need to know, if I have a DNA sequence, what's going to be my mRNA sequence and what's going to be my amino acid sequence. And this all depends if the DNA sequence we're looking at, is this the template strand or the coding strand. Remember the difference. Template strand is what is being copied by the RNA polymerase. Okay, the RNA polymerase attaches to the template strand. Coding strand is what looks exactly like mRNA. So if they tell you that, hey, this sequence is the coding strand, your mRNA will look exactly like this, which means my mRNA sequence will be UAC, UUA, GCA, etc. So I'm just copying whatever's on there. Template strand, that's a different story. You're gonna, instead of U here, you're gonna have A. You're gonna have U here, G here, and so forth. And then amino acid, how do you get it? You look at this. If this is the coding, then you look up UAC. Where's my UAC? I start with here, U, This is my U section. A, C, UAC. So I have tyrosine. So for my first amino acid, I'm going to write tyrosine. Second one, by the way, if this is the coding strand. If it's template strand, you got to do the conversion first. And here, UUA. So UUA is leucine. Next one is leucine, and so forth. You just go down each codon and find your amino acid. So clearly this is the template strand, because they've had to convert from this to this, AUG, and that actually makes sense because AUG, which is my methionine, is always the start. So you usually see AUG as the start. And then you have these stop codons here. So whenever you see these, that's the stop codon. So you do see that here. So this is actually template strand. Your first is always this guy. And then you're going to have AAU, AAU, asparagine. You have CGU, arginine. UCG, serine. Okay. And then GGG. And then the last one is just a stop codon. So this is your final amino acid sequence. So there you go. Methionine, asparagine, arginine, serine, glycine, and then stop. You don't write this there. I don't know why that's there. Okay, now the wobble effect is what introduces redundancy. So the third base pair between tRNA and mRNA is sometimes ambiguous. So this ambiguity causes what? Redundancy in the DNA code. Some tRNAs can recognize several different mRNA codons. Example, the same tRNA can recognize GCC, GCU, GCA, and GCG. So this is alanine. Let me just scroll back up. Which one is this? Yeah, alanine. Okay, so let's look at what this looks like here. We have yeah, this is not what they're saying up here. This is not the right diagram. What is being said up here is that you're going to have G, let's say this guy, CGG, not U, G. It's the same codon up here, but it'll still recognize different nucleotides in this third position right there. because it doesn't directly bond like this. Yeah, I don't like this diagram. Let me see if I can pull up. A diagram for this wobble effect. Yeah, so you see this? You have something coding a tRNA coding leucine. It's the same tRNA. You see GAG, GAG. So this position on the tRNA doesn't change. Here, on the other hand, we have CUC, CUU. They're both recognized by this tRNA. Another example, so wobble refers to how you see this roundness of the tRNA. It actually positions tRNA in this way. So it'll bind directly for the first two, and then the third one is a little wobbly, okay? So it doesn't directly bond, and so it can actually recognize, or it's okay with other nucleotides in that position. So this is another example. So the third position is the wobble position on the tRNA. Again, remember that this tRNA loop, the anticodon loop, is a loop. So it's like half a circle. And so it's not a straight line like the mRNA. So the pairing is not directly straight. So yeah, I don't think you guys need to know more than that. But as I said, this is not how it's supposed to be drawn. So it should be the same. How does tRNA bind to its amino acid? Aminoacyl tRNA synthetase catalyzes a reaction that joins a tRNA to its amino acid. There are 20 types of this enzyme, one for each amino acid. Okay, you guys can see how this happens. It does require ATP, adenosine triphosphate, comes in here. Amino acid is phosphorylated. Once it's phosphorylated, it's in its active form. Now it can bond to something else. The tRNA comes along. It has to be specific to this enzyme. The enzyme is specific to the amino acid. tRNA is specific to this enzyme. And so by specific, I mean it has a very specific sequence. It has to fit just right. So that's why it's specific. The amino acid is specific to the tRNA. Okay, now let's look at ribosomes. This is the site of translation. So ribosomes are made of proteins and ribosomal RNA. There are two subunits, large and small. You can see the blue one. This top portion is the large subunit, and the bottom portion is the small subunit. Subunits and ribosomal RNA made in the nucleolus. So inside the nucleus, there is a specific area called the nucleolus, and that's where the subunits and the RNA are made. And then the subunits are kicked out into the cytoplasm separately. So you won't see both of them attached, walking away, going into the cytoplasm. The subunits only join in function when attached to mRNA. So you'll see them floating around. Why isn't this working? Oh my goodness. Okay. You have your large subunit. Looks like this. It'll float around. You have your small subunit like that floating around. And then once an mRNA strand comes, then you're going to see this sandwiching happening. You have your mRNA. What's happening here? And then you'll have your small subunit and then the large subunit. They come together, sandwich the mRNA, and start copying or translating. Okay, this is the ribosome is different between prokaryotes and eukaryotes. This is another difference you guys have to memorize. The ribosomes are larger in eukaryotes, larger in size, slightly different chemical compositions. What are some applications to this? In other terms, how can we use this to our advantage? When something is different between two organisms, it creates the opportunity to target one of those organisms specifically. But when something is the same between two organisms, if you target that thing, you're targeting both organisms. So an example is if you're targeting a specific amino acid sequence or codon or something, codons are universal. So if you're attacking a specific codon, you are attacking both organisms. But if you have a bacterial infection, you can target things on the bacteria that are not the same in the human. An example here is your ribosomes. So the ribosomes are different between the two organisms. So if we can target bacterial ribosomes, we can kill the bacteria without hurting the human. So how are they different? Let's look at this in detail. How are they different, the ribosomes, between prokaryotes and eukaryotes? The large subunit looks like this. This is what we call 50S. The S is just a unit of measurement. You guys don't have to worry about it. In eukaryotes, it's 60S. So it's slightly larger. And then the small subunit in the prokaryote is 30S. In eukaryotes, it's 40S. Slightly larger eukaryotes. When they come together, I know this doesn't add up. Again, you guys don't worry about like 50 and 30. When you add them, it doesn't give you 70. Just don't worry about that. But for the prokaryote, the full ribosome is 70S, whereas for eukaryotes, it's 80S. You can look at these details. They don't matter. You guys don't need to know this many details. You just need to know these numbers. Okay, so now let's look at inside the ribosome, and let's look at the active sites, the functional sites or areas within the ribosome. So there are three major sites for the ribosome. We have the A site, P site, E site in this order. The A site is the arrival, where the tRNA first attaches. P site is, think of the P for the polypeptide chain. This is where you're growing your polypeptide. You're going to see your polypeptide growing out of here. And then the E site is where the tRNA enters. After it's released its amino acid, it enters the E site for exit to exit the ribosome. Okay, 4 binding sites. The last one is where your mRNA actually inserts itself to be read. Okay, let's look at this in stepwise detail. An initiator tRNA with anticodon UAC pairs with start codon, carries the methionine. So remember, we said this is always the start. You'll always start an amino acid sequence with met or methionine. No, GTP drives this reaction, so it's endergonic. GTP is guanosine triphosphate, so it's an energy molecule. Okay. So looking at this diagram, we have our small ribosomal subunit which binds this mRNA. The tRNA comes, the UAC anticodon tRNA, which complements AUG. This codon codes for methionine. This tRNA has methionine attached. It comes and finds the start codon. As soon as that happens, the large subunit comes, recognizes this, and then binds to it. This is driven by GTP. Okay, so codon recognition. New tRNA binds to the addition site over here. It is based on the next codon. So we have our methionine first, and then the next thing determines which tRNA comes in here and is able to bind. So it comes in there, binds, and then a peptide bond is formed at that active site. So at the P site, this is where we're adding. We put the long chain onto our new amino acid. Once that happens, this body moves to the right. This is now empty. So enters the exit site. This one has the long chain. Enters the P site. Opens up the A spot for another. So you can see this is empty, moves here. This has the long chain, it moves to the P site, has the polypeptide. The A site becomes empty, and now a new tRNA can enter based on the next codon. So this keeps on going until we hit a stop codon, which at that point, this whole thing disintegrates, these fall apart, and you have your full amino acid. Or sorry, full polypeptide. So this is the phase 3 termination. So this is where you reach the stop codon. A release factor binds to the A site. Release factor separates polypeptide chain. And last tRNA ribosome subunits and mRNA separates. So over here, the release factor binds, allows this tRNA to separate from the polypeptide chain. Once it's separated, this thing enters E site, exits, polypeptide chain is released, and this whole thing disintegrates. So, just as we had with our pre-mRNA, where we had these modifications, once we create our polypeptide chain, we also do a set of modifications. So, certain amino acids may be chemically modified by the attachments of, how are we modifying? We can add sugars, we can add lipids, we can add phosphate groups. Enzymes may remove one or more amino acids from the leading end of the polypeptide chain. Two or more polypeptides may come together, becoming subunits. So you have one chain just produced here, another chain produced here. They'll come together and create one protein, which would be the quaternary structure. The definition of a quaternary structure is two or more polypeptides that are attached to each other. Okay, so this is a good list of all the post-translational modifications. Lipidation, adding a lipid. Ubiquitination adds a ubiquitin to a lysine residue, marking it for destruction. This thing, destruction. So anything that has ubiquitin, the cell recognizes as garbage and it starts to destroy it. Disulfide bond is when you get sulfides bonding each other from different amino acids. So this can be tertiary structure. Acetylation, you add an acetyl group to the N terminus, increases its stability. So just like the mRNA 5' cap and poly-A tail, this is similar to that in function. Glycosylation attaches a sugar, usually to N or O atom in the amino acids side chain. So glyco refers to sugar. Phosphorylation adds a phosphate group to serine, threonine, or tyrosine. You guys don't need to know specifically which amino acid we're phosphorylating. Polyribosome. So in prokaryotes, we did say everything is happening in the cytoplasm. Nothing is compartmentalized. And as a result, when we have an mRNA like this, we can get, to speed things up, we can get multiple ribosomes attaching to one strand of mRNA, which means we don't need to produce too many mRNAs to start producing all these proteins. So at any given time, you can start to produce like multiple proteins from just one strand of mRNA. This is a real image, a TEM image of this long strand of mRNA being translated by so many ribosomes. Okay, this cluster of several ribosomes is just called the polyribosomes. Okay, recall protein structure, primary structure, order of amino acids. Okay, these are the primary, secondary, tertiary, quaternary structures. Primary is just the order of amino acids. So these are just the covalent bonds. In like amino acid. So this sequence is just the primary structure. Secondary structure is the coiling or folding. This is the alpha helix or beta pleated sheets. And then you have the tertiary structure, which is like it's wound up. It's not just alpha helix or beta pleated, but it's like a more wholesome shape. It can include SS bonds or disulfide bonds or bridges. By the way, the coiling and folding, these are created by hydrogen bonds. And then quaternary structure is 2 or more polypeptides together. Polypeptides coil and fold immediately. So usually it's the amino acid sequence. The sequence itself determines the shape, the folding of the polypeptide. So if it's, for example, you have a chain, and let's say this section, so this is my amino acid chain, or I'll say polypeptide. Let's say this section of my polypeptide is hydrophobic. Okay? And let's say this section is hydrophilic. So my hydrophobic section is going to tend to aggregate and like fold within itself because it wants to decrease exposure to surroundings because it's water in the surroundings. Like cytosol is water, essentially. And this side is going to want to expose itself to water. So the folding is going to depend on these sort of, whether it's polar, non-polar, hydrophilic, hydrophobic tendencies of these amino acids. So this is going to fold in such a way that it just aggregates within itself because it likes to do that. It's hydrophobic. Okay, so again, let's look at this visually. Primary structure is just your sequence. Secondary structure is either your alpha helix or beta pleated sheet, or none of the above. Honestly, you don't. Some sequences don't create beta pleated or alpha helices. And then you have the tertiary, where you see it's like pretty much coiled up. All the like hydrophobic aggregate next to each other, hydrophilic expose, whatever. And then the quaternary structure, you have multiple subunits. So you'll have this as one subunit, and then you'll have more subunits attached. Free versus bound ribosomes. Structurally, these ribosomes are the same. So you have your 60S ribosome in humans. And then you have your smaller 40S floating around. So structurally, they're the same. Free ribosomes make proteins that will stay in cytoplasm. So you see these cytoplasmic ribosomes that are floating around. Those are making the proteins that are going to stay in the cytoplasm. Because if we want to ship it somewhere, then the protein better stay in this lumen, because that's how we start shipping things, remember? So bound ribosomes, these ones, which are attached to the rough ER. Remember, rough refers to these ribosomes that are attached. It looks like a rough shape with all these bumps created by the ribosomes. So bound ribosomes make proteins that will attach to the endomembrane system. Remember, the endomembrane system is this membrane. We said that it's continuous. This is the membrane that will pinch off, go on to the Golgi, integrate into the Golgi, become part of the Golgi, and then later, again, pinch off, become a vesicle, and then go to my cell membrane, plasma membrane. It goes as a vesicle and then attaches itself Okay, it attaches itself like that, and then kind of opens up, integrates into the membrane like that. It becomes part of the membrane. Okay? So the protein might either be like extracellular, so it's in this space, it's being carried over here, and then it's released outside, or it could be a membrane protein. Let me choose another color here. It could be a membrane protein, which means it is stuck to the membrane. It is integrated into the membrane. So once it reaches the membrane here and integrates in the membrane, it stays on the membrane just like that. So these are my possible options, right? The endomembrane system is the membrane of the ER, nucleus, Golgi, lysosomes, vacuoles, and plasma membrane. Or it'll be secreted. Any questions so far? Okay. So what determines which one it will be? All proteins start off being made in cytoplasm by free ribosomes. If there is a signal peptide, this signals the ribosome and the peptide to move to the ER. So there's a receptor that will bind, well, the signal peptide will bind a protein, which is a signal recognition particle over here. Well, it would be like this one. Attaches to the signal peptide right here, which then helps bring it to the translocation complex, which has an SRP or signal recognition particle receptor. So it has a receptor, it binds this, which is obviously bound to this polypeptide with the signal peptide. Once that happens, this thing opens up and pushes this growing peptide chain into here. So now it's in the lumen. You see that? It starts growing within the lumen. It grows here, and then the signal peptide is actually removed. It was just a signaling piece. So once it serves its function, it's actually removed. And now we have our protein within our ER lumen. Okay. Oh, so these slides are just going over each step that I went over. Signal peptide cut off. And once we cut that off, we release this protein into the lumen. So, protein targeting. We have different destinations for proteins. It's either going to be secreted, so it's extracellular. It's going to go in the nucleus, mitochondria, chloroplast, cell membrane, cytoplasm, etc. Here's a list of all the different types of RNA in a eukaryotic cell. We have our messenger RNA. You guys already know what that is. Transfer RNA, you know what that is. Ribosomal, part of the ribosome. It's what recognizes the mRNA. We have the primary transcript. So this serves as a precursor to mRNA, so basically pre-mRNA or rRNA or tRNA. It will be processed later. Well, this is the primary transcript. Small nuclear RNA or snRNA. This plays structural and catalytic roles in spliceosomes, if you recall. The complexes of protein and RNA that splice pre-mRNA in the eukaryotic nucleus. SRP or signal recognition particle RNA. It's part of that signal recognition particle, which recognizes the signal peptide. So let's go back up here. It's within this thing. So on this side, you're going to have the SRP RNA integrated into this protein. Coupled transcription and translation in prokaryotes. So as you can see, prokaryotes, again, with their doing everything all at once, you have your DNA strand. Long piece of DNA strand. Let me just draw it out so you can see it. DNA is running right there. That's my DNA. Again, prokaryotes. Eukaryotes don't do this. Why? Because DNA is only in the nucleus. Ribosomes are outside. So we actually have to ship our RNA outside to be able to translate. Now from this DNA, we're going to create, so red is, DNA. Let's say green is my RNA or mRNA. So these are all mRNAs. And then I have my ribosomes attached to the mRNA, creating proteins. So let's say proteins are blue. You're going to see proteins popping off of here. And you guys have to know, the shorter side of the mRNA is going to have the shortest protein chain, like so. And then once it moves down, the ribosome moves down, it's going to create longer and longer pieces of protein. Okay. And the other thing you can note down here, if you're given a diagram like this, you could deduct the directionality of your DNA and your mRNA. So you can see that this is short. So it's fresh or new. This is long. So it's basically you know that this RNA polymerase is moving in that direction. Because by the time it's down here, it's produced this whole long piece of mRNA. So if it's going in this direction and it adds to the 3' end of mRNA, we know that this side is 3', this side is 5'. And directionality of DNA is the opposite. So 3' here, 5' here. Like some of these things you guys have to be able to do. Okay, let's look at the differences here. Very important to be able to compare them. Prokaryotes, DNA is in cytoplasm, circular chromosome, whereas eukaryotes, linear chromosome. Naked DNA, for eukaryotes, DNA is wound onto histones, which they can be coiled tightly, so you can't access that DNA, or it can unwind and coil lightly, so you can actually access that DNA. There are no introns in prokaryotes. They don't need them. They need all of their DNA, like all the time. We have introns in eukaryotes. So this is the full picture in a nutshell. So step 1 in protein synthesis is transcription. Once it's transcribed, we have RNA modification. So you see there's alternative splicing. We're going to cut out the introns. We're left with exons. We stick poly A tail and 5' cap. Ship it out to the cytoplasm. Ribosome comes. This last step is translation. The ribosome subunits come. They bind the start codon and the methionine tRNA, and then they start the translation. Well, I guess this is part of translation. Yeah. And then sticking all these amino acids to the tRNA. There are aminoacyl tRNA synthetases that are specific to each tRNA and amino acid. So we have 20 or 21 of them. So basically, you need to be able to describe what I just described if you want to have a good understanding. So in terms of studying, this is what I would do. I'd sit down, start a blank piece of paper. So I'll have a completely blank page in front of me, and I'll start with the first step in protein synthesis, which is transcription. Move on to the second step, third step, and so forth, and draw this whole thing out from memory. And if you need help here and there and you have to look things up, then you know you don't have this completely figured out. You can look it up and then start over. And you do this enough times where you just draw this whole thing from memory, and that's how you know you've learned it. Okay, last thing, we're already at 11. If someone has to go quickly, they can, but I do want to cover mutations quickly. We did start late anyway, so we can go on for another 5 minutes. So, mutations. Genetic variation and mutation play roles in natural selection. A diverse gene pool is important for the survival of a species in a changing environment. Alterations in a DNA sequence can lead to changes in the type or amount of the protein produced and the consequent phenotype. We can have good mutations, we can have bad mutations. DNA mutations can be positive, negative, or neutral based on the effect or the lack of effect they have on the resulting nucleic acid or protein and the phenotypes that are conferred by the protein. So many things to consider here. I'll go over it, I guess, in detail in this section, but as you can see, you have to look at what's the nucleic acid that results? What's the protein that results? So the amino acid sequence. What's the phenotype that results? So you have to consider all these things to determine, is this positive, negative, or neutral? Errors in DNA replication or DNA repair mechanisms and external factors, which include radiation, reactive chemicals, like smoking, for example, can cause random changes or mutations in the DNA. Whether or not a mutation is detrimental, beneficial, or neutral depends on the environmental context. Mutations are the primary source of genetic variation. Example right here, the environmental context example, is let's say you have a bunch of bunnies in an area that is very snowy. And there is a mutation in the bunnies that creates white fur. So it knocks out the pigment production in the fur of these bunnies. It's a mutation. Basically means the pigment production stops, and so the bunnies actually have white fur. Is this a positive mutation? Is it beneficial? Or is it detrimental? Or is it neutral? We consider this beneficial because now the bunnies can blend into the environment. They won't be detected by predators, so they can live longer. We consider this beneficial, even though it's a mutation. So that's one example. Let's look at the different types of gene mutations. We have something called a frame shift. One nucleotide is inserted or deleted. The moment you insert or delete a nucleotide, as you can imagine, you are shifting the sequence of codons after that insertion or deletion. So for example, here we've removed a U. Once you remove the U, instead of U being here, and then G, C, and so forth being there, you've removed the U. This was supposed to be U, coding for phenylalanine. You've removed it, so now what's the result here? The result is UUG, which actually codes for leucine and not phenylalanine. So that resulted in a change of, always, insertions and deletions change the amino acids after that mutation is made. So anything beforehand or upstream, it's okay because it doesn't shift the frame for those ones. But anything after the mutation, we're going to change the amino acids. So that's a frame shift. Silent mutations. There's no amino acid change. Why? Because there's that redundancy we talked about. So it's not included in one of these diagrams, but if you change that third amino acid in a codon and it still codes for the same amino acid, it is a silent mutation. It's still a mutation. It's considered a mutation. It's just silent. So this is considered a neutral mutation because there's no change. Frameshift mutations, you have to look at the end product, the phenotype, to determine is this positive mutation or negative mutation. We can't know just by seeing if it's frameshift or not. Okay, if we have a frame shift, we have two types. Either missense, which changes amino acids, or nonsense, which introduce an early stop codon. So in this case, this frame shift has caused an early stop codon. The stop codon may have been down here initially, but now we've introduced an early codon, stop codon. So that's called a nonsense mutation. So as you may imagine, if you insert or delete one full codon, then you're not actually changing the amino acid sequence, because there's no frame shift in that scenario. A more common type of mutation is a point mutation, which is where one nucleotide is substituted for another, which results in not necessarily. You don't have to change the amino acid. Because of the redundancy, sometimes the amino acid doesn't change. So this is one example showing you how sickle cell is formed. It's a point mutation going from glutamine to valine. We'll talk about that later. Oh, we can talk about it now. Okay, so in sickle cell, the reason why you see these sickle shapes in red blood cells, and it's not this like plump donut type shape, is because in position 6 of our amino acids, we go from glutamine, which is a polar molecule, to valine, which is non-polar. Non-polar means hydrophobic. Which means these valine residues now in the mutated red blood cells or sickle cells, they start gathering towards each other. So this hemoglobin now has a valine in this position that it's not supposed to have. So it's going to, the hemoglobin for my next protein, so the next hemoglobin also has a valine here. It's hydrophobic, so it's going to aggregate with this valine. I'm going to have another hemoglobin molecule with a valine here. It's going to aggregate with these valines. So we have all these hemoglobin molecules aggregating in this position. And once it does that, it creates this sickle shape. And it can't carry oxygen properly. So that's how you get the disease. And that's it for this set of slides. Any questions for you guys?
Can you quickly talk about polyribosome again?
The prokaryotic situation? Yeah, here. So basically, as soon as you have a strand of mRNA, you can start attaching ribosomes. So you would actually attach it right here. The ribosome would move up, and then you'd attach the next ribosome, and it would move up, and so forth. So this would be the oldest ribosome. It's about to pop off, and it has a long protein chain. And then this one has just a little shorter protein chain, shorter, because it's more fresh. Short. And this is just starting out the protein chain. So one mRNA strand, but you'll have multiple ribosomes attaching to it and moving forward and creating protein. And you don't stop with one round. You keep attaching ribosomes as soon as space becomes available, and you keep producing more and more proteins. As you can see, you can create a lot of proteins with that system. Any other questions? Does this make sense?
Wait, so will all of these proteins be connected at the end later? Or they just do different functions?
Not necessarily. They won't do different functions. They're all the same function because they're the same protein. They have the same sequence, so they're essentially the same thing. So they have the same function. But
Okay.
they don't necessarily connect with each other. They're just like the cell is, it doesn't just require one protein to go and do, a bunch of enzymatic reactions, for example. You need a ton of enzymes to be able to do a reaction quickly, right? So you're going to need a ton of proteins. And you can
So
get
what
them.
about eukaryotic situation? Like you said, this is prokaryotic, right?
This is mostly prokaryotic.
Okay. I got it.
Okay. But the main thing with prokaryotes is that transcription and translation happen at the same time. Eukaryote, so what you need to take away from this is transcription and translation in prokaryotes happen at the same time. You see the RNA polymerase. RNA polymerase right here, creating mRNA, and then ribosome creating protein. So this is transcription. This is translation. They're happening at the same time. In a eukaryote, this does not happen. You do transcription, ship out the mRNA, and then do translation. That's the whole point of this slide. Okay, other questions?